How's it going, you guys? So in today's video, I wanted to kind of distinguish between some terms that students oftentimes get confused with. That is chromatin, chromosome, chromatin, centrosome, centromere. So a lot of these terms sound very similar. So in this video, I wanted to distinguish between these terms to hopefully clarify the difference between them. So let's first start off with the term in the upper left-hand corner of the board. This term is DNA. So DNA is our genetic information. It has this sort of double helix structure. Some might refer to it as a twisted ladder-like shape. So that's DNA. For this particular unit, we're not going to go in depth with DNA. That's going to be later on. But just note, DNA is our genetic information. It controls our characteristics, our phenotype, if you will. And yeah, DNA is a double helix, and it's a long molecule. So I'm going to put ... on either side of the DNA. So this is a zoomed in version of DNA. So imagine if we zoom out a little bit. What we're going to see, I'll still put a ... but now I'm going to represent DNA as more of a line. And every now and again, I'll make this a little bit bigger actually. Every now and again, DNA is going to be wrapped around these protein complexes called histone proteins. So I'm going to draw it as a circle with a kind of cross dividing it into 4 sections. So this right here is DNA, and inside of where the DNA is wrapped around, we have histone proteins. Histone proteins. Sometimes just called histones. Histones, if you were to look at them alone, are usually found in a group of 8 proteins clustered together in almost a box shape. So you can imagine 4 histone proteins like this, and then another 4 proteins right behind them, making kind of like a box. So histone proteins have a very specific purpose. I'll put ... over here because DNA, again, is a very long molecule. But histone proteins serve the purpose of helping the DNA to condense during cell division. So I want you to imagine, I'm going to grab my laptop charger, but my laptop charger can kind of represent DNA and histone proteins and how they work. So if you were to imagine the cord of this charger being DNA, and then if you were to imagine this central box being the clump of histone proteins, at any given time, DNA is going to be wrapped around this clump, like so. So in this situation, it's kind of loosely wrapped, right? We have a lot of exposed DNA. But during cell division, if the cell needs to divide, we need to condense those chromosomes, or which I'll get to in a minute, condense the DNA. So we will wrap the DNA around the histone proteins, even tighter. And as you can see, that's going to allow the DNA to condense. So that is the role of the histone proteins. It's to allow the DNA to condense when it needs to. So I'll put this over off to the side for right now. Marker. But that's it for chromatin. So chromatin is basically just DNA wrapped around histone proteins. Now, moving out, or if we kind of zoom out from chromatin, and we kind of get the whole DNA molecule, now DNA molecules are pretty long, but get the whole molecule in view, that is a chromosome. So let me draw that over here. A decondensed chromosome looks like this, where we zoom out even more. Every now and again, the DNA is wrapped around histone proteins. And this is DNA in its decondensed form, so it's loose. This will not be drawn to scale necessarily because I cannot picture the size of DNA, or I cannot depict that. on my little space of whiteboard because it's a very long molecule. But if you could imagine that the chromosome, or that the DNA, we have zoomed out completely from the DNA so that we see one end of the DNA, and over here we see another end. And throughout the process, every now and again, we have DNA wrapped around histone proteins. That could represent a chromosome. So a chromosome, I would define it loosely as a full-length strand of DNA with its associated histone proteins. So that is the loose definition that we'll give it for right now. Now, I want to grab a slightly better marker. Here we go. So that is a chromosome. So we can label the line, the DNA, and we can point to this little bundle here and label that histones. Again, histones and histone proteins, same thing. As for the chromosome itself, if we are talking about human chromosomes or human cells, in any cell, within the nucleus of any human cell, we have a total of 46 chromosomes. So that means we have a total of 46 full-length strands of DNA. Now, imagine, if you will, that we take this DNA and we wrap it even tighter around those histone proteins. So you can imagine that the DNA molecule is going to start to condense. So if we condense it, we'll see something like this. The DNA is wrapped around those histone proteins, causing the entire molecule to shrink and condense. And what we see is something like this, DNA in a bar-shaped fashion. So I would say this is a bar-shaped chromosome, and I'll put condensed. So if we condense the decondensed chromosome, the loose chromosome, we get a condensed bar-shaped chromosome. So a bar-shaped chromosome, if it only has one condensed DNA molecule, we say it has one chromatid. Chromatid. Different from chromatin. So chromatin is DNA plus histone proteins. A chromatid is a single condensed bar of DNA plus histone proteins. So that is chromatid. And for every one chromatid, we have one DNA molecule, full-length DNA molecule. All right? Now, I want you to think about when cells divide, cells can't just keep their 46 chromosomes and just divide like that, because then we'll have 23 in this cell, 23 in this cell, and then if they try to divide again, we're slowly losing DNA. So you have to imagine that the DNA has to double, or the whole cell needs to double everything within the cell before it divides, and that includes the DNA. So the DNA has to double before we divide, so that both of the daughter cells, the ending result cells, are going to have the same number of chromosomes as the parent cell. So what happens is if we take this DNA molecule and imagine multiplying it by 2, what we get is an X-shaped chromosome. So an X-shaped chromosome has 2 chromatids, 2 DNA molecules that are condensed, and I'll draw that up over here. So you can imagine 2 chromatids, 2 DNA molecules that are condensed, and you can kind of see that they're bent so that the center of it are kind of close together. They are connected by what is called a centromere. So let me label that. This right here we'll call a centromere. Technically, we can say that there is a centromere here as well, but it serves less of a purpose on a single chromatid, a lone chromatid, a bar-shaped chromosome, and it serves more of a purpose for the X-shaped chromosome. So 2 chromatids stuck together is an X-shaped chromosome. This is where students kind of get confused because at this point we've only had one DNA molecule. Here we've only had one DNA molecule. Here, even though we have two DNA molecules, two chromatids, it's still considered one chromosome. So it gets a little confusing. And Lord knows why they named this a chromosome and this a chromosome, even though this has double of the genetic information, double of the DNA. But it is the way it is, so we'll just continue to roll with that. If we see two chromatids stuck together like this on an X-shaped chromosome, I'll point to these two chromatids. We don't just call them chromatids, we call them sister chromatids. Chromatids. So we have our sister chromatids attached at the center through a centromere. And on either side of the centromere, I'll put something like this. On either side of the centromere, we have these things called kinetochores. So kinetochore. It's kind of spelt like kinetochore, but it's a hard k sound. Just like you wouldn't say chromosome, you would say chromosome. Same thing here. We have a kinetochore over here. Kinetochore. So we have a kinetochore on either side of the centromere in an X-shaped chromosome. And the purpose of the kinetochore, it is a protein complex that serves as a binding site for these things called spindle fibers, which we'll talk about. Spindle fibers attach to the X-shaped chromosome on either side and then pull to rip the chromosome apart into two bar-shaped chromosomes, two individual chromatids. And so when it pulls and it rips it apart, it is attached through the kinetochore. The spindle fibers are attached to the kinetochore. So we'll talk more about that. It'll make a little more sense once we go into the steps of cell division and the cell cycle. But those are some terms that students oftentimes get confused, and hopefully this demystified some of those terms, made them a little more understandable. Now the last term that I left out over here was centrosome. I left that out initially because it has a little bit less to do with this whole thing. It doesn't include DNA, which is why I left it out. But it does sound, centrosome, kind of sounds like centromere, so I wanted to put it up there so that we can distinguish between the terms. A centrosome is a pair of centrioles. So I'm going to depict that as two rectangles. So I'll say this is a centrosome. This right here is one centriole. So a pair of centrioles is a centrosome. So centrosomes are responsible for being the starting points of spindle fiber production. So spindle fibers are basically built off of the centrosomes during cell division. So we'll talk more about that again when we talk about cellular division, mitosis, prophase, metaphase, anaphase, telophase. So we'll talk all about that soon enough. But these are all the terms that students get confused during this unit. So hopefully this video helped. And if you have any questions, let me know.